Okay. That's a Lenovo computer right there. It's a fellow Lenovo webcam cover.
Okay.
Okay. All right, so I'll start the webinar in a few seconds.
Perfect. When I see you going, closing your screen, I will know that we are going to start recording, and I will start with a brief introduction just of the session, and then we jump right into it.
I'm going to start the webinar in 10 seconds. For the first like 1 minute, let's not get to the presentation right away, because people might have some delays connected.
I will remember about the chat, this kind of thing.
All right, going live and forward.
Thank you.
3, 2, 1, start.
Hello, everyone. It's right at 9 for me here on the East Coast. So I'm Valentina, and together with Taha, my amazing helper behind the scene, we bring to you 3 great speakers this morning. Sophia from Indiana University, Estratia from the University of Nottingham, and Margaret from the University of Oxford. They will each have 12 minutes to present their work, followed by 3 minutes for your questions. And please use the Q&A option at the bottom of your Zoom screen to type your questions during the talk, and I will have the pleasure of reading them to our speakers right at the end. And from time to time, do check the chat, because we will be posting links and references to our speakers' talk. And with this, it's my pleasure to leave the stage. Let me put this. It's my pleasure to leave the stage to Sophia. Sophia Vinci Boer, a postdoctoral fellow in Dr. Franco Pestilli lab in the Department of Psychology at the Brain and Science Indiana University. And she'll present us her work on the developmental differences in white matter tracks between and within the dorsal and the ventral stream. Please, Sophia, take it away.
Hi, so thank you for the introduction. I'll be presenting findings from a recent study that investigated the development of white matter contained within the dorsal and ventral streams, and also a white matter pathway that directly connects those 2 streams. It's widely accepted that visual information is processed along at least 2 pathways. There's the ventral and the dorsal visual pathways. We know quite a bit about white matter development in these 2 pathways. For example, we know that the tissue properties or the microstructure properties of this ventral stream here are adult-like by the age of 5, whereas the microstructural properties of this dorsal pathway undergo a more prolonged developmental trajectory, not reaching adult-like values until basically adulthood. So recently, a set of white matter tracks that connect the ventral and dorsal pathways have been rediscovered. So we are calling this the posterior vertical pathway. This posterior vertical pathway connects ventral and dorsal pathways directly. And we know essentially 0 about the development of this pathway. So one of the goals of this work was really to understand more about the microstructural development of this posterior vertical pathway, because it has the ability to really communicate directly between these ventral and dorsal visual streams. We were particularly interested in understanding how the development of the posterior vertical pathway, that I'll refer to as PVP, that I'll start using that terminology, how the PVP was related to the development of the dorsal and ventral white matter. The location of the PVP here seemed to suggest to us a particular feed-forward flow of sensory information. And there's a lot of work showing that sensory input or sensory flow is a major driver of developmental change. So we expected that this feed-forward flow might have some effect or some relationship to the order in which white matter tracks develop. So this model posits that some portion of the microstructural development is based on just receiving and carrying information. So the sensory information if sensory information were to follow the white matter architecture in a feed-forward fashion, it would first start out here in the occipital cortex along the vertical occipital fasciculus here. It would then travel down the ventral visual stream along the ventral horizontal pathway. And then it would travel up to the dorsal stream along this PVP pathway that we're particularly interested in here, before traveling along the dorsal horizontal pathway and then along pathways in frontal cortex, for example, the frontal slant here that I'm depicting. So we started with a simple hypothesis. We hypothesized that the microstructure of the vertical pathway develops after the ventral pathway and before the dorsal pathway. And then we kind of tacked on the VOF and the FAT here to kind of get, to bookend the three pathways there. We collected diffusion MRI data from children ages 5 to 8 years and an adult comparison sample so that we could understand the relationship among the three pathways in development, but also in adulthood. And in brief, we first identified each tract and pathway using diffusion tractography. So here I'm showing you an example of a child and an example of an adult. And here are those same, the VOF, the ventral horizontal pathway, the PVP that we're very interested in, the DHP, and then the FAT here. For each tract, we computed the microstructural property, in this case, fractional anisotropy, or FA, along each tract here. So here I'm showing you the ILF, and this is the tract. And we computed FA just along that tract here. So this is the mean for the adults, and this would be the mean for the children along the tract. We averaged across that tract profile to get the tract mean for the adults and the children, and then we averaged across tracts within a pathway to get the pathway mean. So this would be the mean for the ventral stream, the ventral horizontal pathway. We did the same thing for the other pathways. So here's the dorsal horizontal pathway. That pathway is comprised of the SLF1 and 2 and the SLF3, for example. These are the same types of plots. And then here I'm showing you the means for that pathway with the adults and then children. All of these data were processed on brainlife.io using open web services, and these apps specifically. I'm not sure if this slide will be available after the talk, but you could definitely email me to ask for these specific links to the data so that you could reproduce the results if you like. All right, so we performed repeated measures ANOVA with 2 factors. The first factor was pathway here. So these are the pathways that I've been talking about. And then the second factor was age. We have adults and children. And the dependent variable was FA, based on the tract profiles approach that I just described. And we entered sex as a covariate of no interest. So this interaction was significant. The difference in fractional anisotropy between children and adults was dependent on pathway. And specifically, the age difference here, it increased linearly from the vertical occipital fasciculus onward to the frontal tracts here, in the way that we would have predicted if this feed-forward model of development has any sway or is correct at all. So the PVP here was the first tract to demonstrate a difference between children and adults, which is consistent with prior literature that has demonstrated that this ventral tracts here are adult-like by this age. So we decided to focus here more specifically. We wanted to look at how because the PVP was really the first tract that showed a developmental difference there. So just to find more evidence for this data-driven model, we expected that PVP microstructure should be more similar to, so that this microstructure should be more similar to the ventral horizontal than to the dorsal horizontal pathway. Because if this is mature, then it's sending mature signals through this pathway that can have a developmental effect. To look at that, we computed pairwise correlations among tracts and performed multidimensional scaling on the correlations followed by k-means clustering. So here, for example, this square is showing you the correlation between the FA and IFOF and FA and SLF1 and 2 in the child sample. And for the k-means clustering, we set k equals to 2 to test our hypothesis that the microstructure of this PVP pathway would be more similar to the ventral than the dorsal horizontal pathway. And this is what we found. You can see here, this is the first and second dimension from the MDS. And we find that the ventral stream does cluster, or I'm sorry, that the vertical pathway does cluster with the ventral stream and not the dorsal stream. So finally, we wanted to assess any relationship between behaviors and the microstructure of the PVP. To do this, we performed a multiple linear regression where we regressed composite measures of visual motor skill, fine motor skill, and literacy on FA in this posterior vertical pathway. And we found that PVP microstructure was predicted by visual motor skill. So as visual motor skill increases, so does FA in the PVP, and also by fine motor skill. As fine motor skill decreases, fractional anisotropy increases in the PVP. So our results not only provide support for our data-driven model of white matter development, but they also suggest a key role for the posterior vertical white matter pathways in the development of the dorsal stream. Our main findings are that the PVP microstructure is less adult-like than the VHP and more adult-like than DHP microstructure in 5 to 8-year-old children. We also found that PVP microstructure is more similar to VHP than DHP microstructure in children. And this PVP microstructure did correlate with behavioral measures of learning in children. So we interpret these results to suggest that the development of the dorsal stream trails behind the development of the posterior vertical tracts and may be driven by input received through those posterior vertical tracts during learning. So in the same way, we could say that the posterior vertical tract development trails behind the ventral stream development and may be driven by input received from these ventral stream tracts. These results in our interpretation are consistent with a large body of work that has demonstrated that sensory input is a major driver of developmental change in the brain. And they provide support for this feed-forward model of white matter development. White matter develops earlier in tracts that are closer to sensory input and later in tracts that are further from sensory input. Okay, so this is just my thank you and acknowledgement slide. So thank you to my postdoc advisor and all of my funding sources and all of the research assistants that's helped with data collection.
And thank you for the great talk. Very, very extremely interesting. I will be monitoring the Q&A. We have our 3 minutes. Good, great timing. Okay. We also share the link to your website, to the lab website, so people will be able to check out the software. If I may start, I do have a question already.
Yeah, sure.
You mentioned having sex as a covariate in your analysis. Mm-hmm. And I wonder whether you did find any effect of that. Like, is there any difference in development between female and male?
Yeah, it's a great question. It's there's actually a lot of research showing that there are sex differences in white matter development, for sure. So it's a covariate that absolutely needs to be in there. That being said, in our sample, we actually didn't find any. It was not a significant predictor, and it didn't interact with any of the other predictors that we have in the models. Yeah.
And then I have another probably naive question that comes from my bias in studying language. I wonder whether you looked at differences between left and right, because we know that the development of the visual warframe area on the left side of the brain is, I wonder if that could play a role in.
Yeah, definitely. So absolutely, we actually did do a hemispheric analysis of hemispheric cohesiveness, and we found actually that in the children there really wasn't any reason to separate left and right tracks, so we grouped them together. So that was true in the first 2 analyses. So in the ANOVA analysis, where we were looking at the developmental progression of tracks, and then also in the secondary analysis with the clustering, it didn't really matter if we looked at the left hemisphere, the right hemisphere, if we put them together, if we, it didn't seem to matter, but it did matter in the behavioral correlations. So when we really kind of dug deep into, I just presented sort of the big blanket result there. So that the result that I presented was that the posterior vertical pathway at large correlates with visual motor and fine motor skill, but not literacy, which was interesting to me. But when you kind of separate apart, you know, which track specifically was it, or was it the left hemisphere or the right hemisphere, or, you know, and it actually is, that correlation is driven by the left posterior arcuate being correlated with the visual motor composite score.
Interesting.
Yeah, so it showed up in behavior, but not necessarily in the developmental progression. Right.
Also, the other speakers, if they have questions, they can feel free to jump in or write in the chat. I do have one last from my side, a technical question. So you mentioned the software, and I confess I didn't check it out yet, but does it mean that the tracks are extracted like automatically, or is the painstaking job brain by brain to extract the tract? And how this square between adults and kids, like, is it harder for?
Yeah, that's one of the strengths of the lab that I'm in right now. They this brainlife.io platform does a lot of things like that automatically that are and it helps with reproducibility, so that somebody could just go, you know, like, I didn't do it by hand, but a lot of these segmentation approaches that are on there and that are being used broadly have been validated against hand segmentations and things like that. But yeah, so it was automatic, and there was not, we did a lot of work to make sure that it was applicable to the children, and it seemed to be just fine. So yeah.
I will have to check it out, because I know my colleagues are trying to do DTI work on kids, and it's just difficult, and they never agree with each other.
Right. I mean, and it can be computationally intensive, all of these things, and BrainLife really does handle all of that very, very seamlessly. So yeah, I would definitely recommend it.
Thank you very much, Sophia. We have learned a lot.
Yeah.
So if there are no other questions, I think we can move to the second talk right on time. And our second speaker from the University of Nottingham, it's Ratia Paputstelu. She's a PhD student working under the supervision of Douglas Artley, and she will be presenting us FNIR's study on language processing across childhood and adolescence. Thank you, Stratia.
Hi, thank you, Val. Can you guys see my PowerPoint? Yeah, you can, okay, great. Okay, so yeah, hi everyone, I'm Thea. Thank you so much for tuning in. I know there's a lot of talks happening at the same time, so I appreciate that. And as Val mentioned, I'm a third-year PhD student at the University of Nottingham, and mainly my research aims at identifying neural markers of language processing across childhood and development and adolescence in a disorder called developmental language disorder using functional near-infrared spectroscopy. And today I'm going to talk to you about the first part of this research, looking at typical language development using the same technique. So to put in context, before we go on, I'll give you a little bit of background on developmental language disorder. So DLD is a lifelong condition that affects about 7% of the population. And at least for now, we don't know exactly the biological causes behind it. People with DLD present a constellation of deficiencies, both in language production as well as comprehension, and they have very variable phenotypes. So clinically, it's really hard to identify them, especially given that right now we don't have an objective diagnostic tool. So what I aim to do with my project is to use neuroimaging to describe the cortical patterns of activation in these children, and ultimately develop a diagnostic tool by identifying distinctive neural markers in their brain activity when they're processing language. So to do that, the first thing we need to do is find out what typical language development looks, so we have something to compare it against. So what we know and what we don't know about typical language development. We know already that the language network develops both functionally and structurally throughout our lives. Left language lateralization is actually established very early on. Some studies have found it present at birth, or it emerges shortly after. And then during adolescence and childhood, we develop more focused activations within the left hemisphere, so between left frontal and temporal regions, and the number of connectivity between the hemispheres, so between left and right, decreases. And then in adulthood, we observe a shift in activation from frontal to posterior regions within the left hemisphere. And that probably is because as we become more proficient language users, we use more lower level processing regions, which are located in temporal areas, and we reserve frontal regions for more complex tasks. So let's say if you're learning a second language, you would be engaging more brain areas because you need more effort. And what we don't know, however, is when that shift actually happens. And that would be very important if we were trying to determine if somebody has typical language development for their age. So that's what we set to find out, and we hypothesized that if this shift in neural activity has not already happened by the time children reach adolescence, we would observe activity both in frontal as well as temporal brain regions. So how did we do that? We used a neuroimaging technique called functional near-infrared spectroscopy, or FNES, and FNES relies on the same principles as fMRI, but instead it uses light refraction to measure changes in concentration of oxygenated and deoxygenated hemoglobin in the blood. It has been reliably used in the past in loads of language and auditory studies, and also it's been proven quite useful with pediatric populations. Other advantages include that it's acoustically quiet, it's non-invasive, so as you can see in the picture, it's literally just a cup that sits on the brain, and it's robust to movement artifacts, allowing for overt language production. However, on the flip side, it has quite a poor spatial resolution, the order of about 1 centimeter, and poor depth sensitivity, which is about 1.5 centimeter. Depth sensitivity is not really an issue with our study because fortunately the language networks are located on the outer layers of the cortex, so we were still able to access them with FNES. So what we did, we recruit 18 adolescents between the ages of 11 and 16 and 12 children between the ages of 6 and 10. And we screen them to make sure that their hearing and their linguistic and cognitive development were within a normal range. Then we asked them to complete a series of language and control tasks while we were measuring their brain activity with FNES. Language tasks were things like repeating, listening and repeating sentences, or listening a sentence and having to decide whether it's grammatically correct or not. And the control task was mainly them hearing the same sentence again, but this time reversed. So it sounded like language, but did not make any sense. And we measured activity bilaterally from frontal and temporal regions, both during language processing as well as rest. So we did collect some data on resting state functional connectivity, but we haven't had time to analyze those yet. So I'm not going to be talking about them later on. Oh, and our predefined regions of interest are here. You can see them here circled in black. So we had 4 of them, 2 on the left and 2 on the right. So what did we find? So here is the block average hemoglobin time courses derived from our regions of interest at group level. So it's both adolescents and children grouped together. And as you can see on the top panels here, focusing on the red line that represents oxygenated hemoglobin, we had higher activations in temporal regions compared to frontal, whereas if you remember earlier, language is strongly lateralized to the left. So we sort of thought that we would see higher activations on the left hemisphere. So the 2 left panels, as I see them, yeah, you see them the same way, yeah. So we were a bit surprised by that. And we think that that's mainly because all of our stimuli were auditory. So they preferentially activated auditory processing regions, which are in the temporal lobes. Also, as I said earlier, we tend to be using a lot of our frontal regions when tasks are more challenging. And we think that even for our younger children, the tasks weren't that difficult. So maybe that's why we don't see a spike on the left frontal area. But what was most important for us was to actually see whether there were any group differences. So we did a mixed ANOVA, and we found that there was a statistically significant interaction between hemisphere region, that being temporal or frontal, as well as age group. And when we did the further analysis with the simple effects, we saw that the only difference existing was that adolescents had higher activity in left temporal compared to left frontal regions. And you can see these are these green bars over here. And we found no other differences. There were no differences in the children's group. And what does that tell us? So first of all, we were happy to say that FNERS was actually an appropriate tool to track down these maturational changes, because they've used it before with children, but never comparing two age groups that were so close to one another. So we were happy to at least have a difference there. And adolescents, as we thought they would, they had more adult-like brain patterns, showing higher activity in left temporal areas, perhaps reflecting the automation skills that is not yet present in younger children. But about that last thing, for us to be able to say that 100%, we needed to find a significant interaction between age group and region within the left hemisphere, rather than having both hemispheres there. So we think that's because of our very small sample size. So the children's group was only 12 of them. And yeah, I would strongly recommend in the future, if we could do this again with larger sample sizes, and if it could be longitudinal as well, that would be even better. So following the same children across time. So where does this take us next? Because we're trying to say whether we can apply this to DLD. So we know already that some children with DLD have atypically responses when processing language in frontal and temporal regions. So we want to better characterize those using FNERS, and ultimately we hope to be able to identify differences in different developmental stages in DLD, and also differences between children with DLD and typically developed children. And if we achieve that, maybe we can develop an objective diagnostic tool for the accurate and early identification of DLD that would give the access to more timely interventions. A lot of behavioral studies have shown that the earlier you can access therapy, the better your future outcomes can be, because obviously language is one of those things that has a critical period of development. So you want to catch it as early as possible. So yeah, that's all from me. Thank you so much for listening. Here's my team back in Oingham. And if you want to find more information about DLD, the Ragel organization is a non-profit that tries to raise awareness, and their website is a great source of information.
Thank you very much. Very interesting. We already have some questions coming in, and people should keep writing and upvoting the questions they're most interested in. And I'm going to start reading them. So Aparna is asking whether you could elaborate on your screening method.
Sure. So first of all, we wanted to make sure they have normal hearing, because obviously we would play to them a lot of auditory stimuli. So we tested, it's standard in the UK, you do sort of a run through frequencies, and you ask the children to press a button if they can hear it. So we made sure that everybody could hear above 25 dB, and then we assumed they had normal hearing. And then the language and behavioral assessments was a battery of standardized ones that have cut points defining whether you're normal or not. So we used, I don't know if they are international, we used one called WASI, it's the Wessler Scale of Intelligence, which is adapted for children. And it has things like matrices. It's like puzzles that you need to find which piece is missing.
Right.
Building patterns from blocks. So you give them 9 blocks, show them a pattern, and ask them to arrange the blocks so they fit the pattern. And then language ones, we did the TROC, which assesses grammar. We did the NMC that assesses phonological awareness. And we did one more. We did vocabulary. So we asked them to give definitions to words. And all of them were within normal limits, but we also asked parents to do one called checklist for communication for the parent, which
Interesting.
is used a lot in autism as well. And the parents just judge how their child communicates.
Okay. Just quickly, a partner has a follow-up. Did you try visual language processing also?
No,
Like,
I
okay.
don't. If with visual language processing, she means if we've had written words on the screen,
Yeah.
we try to avoid that as much as possible
Okay.
because we didn't want to add extra workload, especially for the younger ones that were 6. Maybe they weren't proficient
Right.
readers yet, so we didn't want their performance to be affected by their speed of reading. And also, we couldn't have an experiment that's like 3 hours long. So we tried
For
to
children.
because that was actually an issue. Like our paradigm was actually very long. It's about 2 hours, and a lot of kids were getting tired. So yeah.
So the next question is from Andres von Schlenden, and he's asking, even though your focus is on children, adolescents, do you know anything about the time course of the shift to posterior regions in adulthood? So is it a process that it finishes in adulthood, or it actually continue developing into older age?
So when they've done adult studies, at least the literature I'm aware of, they don't really do, they don't sort of track them. So you would get, when you say adults, it would be 18 to 50 years old, and you would just take them as one group without looking into finer differences. I know in older age that these networks tend to become less well connected. I don't know exactly what happens with this change, whether you start engaging your frontal regions more. I know they've done some very interesting studies with second languages, so with adults learning second languages, and then you see that the more proficient they become with a language, then you see this shift again. So I would answer that it is something that develops and changes because it depends literally on how much effort you put. If something is easy and automatic, you wouldn't need as much brain power, let's say.
Makes sense. We have time for one last question, and even this one asks you to speculate a bit. Joao Araujo is asking, what do you hypothesize would happen to the hemodynamic response function in parietal areas, let's say the left and the right IPG, that are usually implicated in integration processes for language tasks across development?
That's actually a very, very interesting question. The problem with that and with FNS is that, as I said, it has quite a poor spatial resolution. And some of these parietal areas would be too close to the temporal areas. So we wouldn't necessarily know whether the activation we're saying is due to the auditory stimuli or whether it's something distinctive. And that was also an issue we had with our frontal, as we weren't sure we would be able to say something separate. And because kids also have smaller heads, the regions are closer together. I know it sounds like a silly reason, but it's actually true. The smaller the head, the harder it is to sort of have this special resolution. But I think it's a really good question. And I think with FNS, the good thing is it's a very fast developing technology, and the spatial resolution becomes even better and better and better. And hopefully soon we'll be able to answer things like that.
So we will all stay tuned and follow you on Twitter to know the next results from your study. Thank you very much.
Thank you.
We can move to our final speaker for this session, Margaret Jane Moore, a PhD student at the University of Oxford under the supervision of Nell de Meyer, and she'll present us a voxel lesion symptom mapping study illustrating how right and left neglect are not anatomically homologous. Thank you, Margaret.
Hi. So as mentioned, my name is Margaret, and I'm working on my PhD at the University of Oxford. Most of my research has to do with studying spatial attention from the cognitive neuropsychological perspective. And today I'm going to be talking about a project we have recently finished looking at the anatomy of neglect. So for those of you who aren't familiar with visual spatial neglect, Visual spatial neglect is a common neuropsychological syndrome characterized by consistently lateralized spatial attentional deficits. And although neglect is generally characterized as just a hemispatial deficit, there are also multiple behavioral phenotypes that you see in the clinical environment. And the two I'm going to focus on today is egocentric, which is self-centered neglect, and allocentric, which is object-centered. And this is an example of what this looks like on a classic neuropsychological test. So in this test, patients are asked to go through and cross off all complete hearts while ignoring the incomplete ones. And patients with egocentric neglect will go through, cross off all the hearts on one side, while patients with allocentric will cross off hearts throughout the whole search matrix, but they'll also make these consistently lateralized false positive errors, which you see highlighted in red. So although these two conditions do frequently co-occur, there's a lot of evidence suggesting they are actually doubly dissociated, and a lot of this evidence comes from looking at the brain. So previous research has done a really good job of characterizing the anatomy of left egocentric and allocentric neglect following right hemisphere lesions. And this is from a really nice meta-analysis done by Magda Czechblocks, which looked at 1,300 patients and found these really nice dissociated different correlates for egocentric versus allocentric neglect impairment within the right hemisphere. And this is quite a nice finding, but as you'll see, it's just within one hemisphere. And if you are familiar with clinical neglect at all, although you pretty much always say, oh, right neglect, right hemisphere damage, left neglect, it actually happens the other way too. So you have a good amount of patients with left hemisphere damage and right neglect, and the anatomy of those patients isn't nearly as well understood. So there's been a couple studies looking at the anatomy of neglect following left hemisphere lesions. This is one that comes from Karnarth. He looked at 11 patients with right egocentric neglect and found that the areas which were significantly associated with right egocentric neglect were kind of the anatomical homologs of the areas that had been previously found to be associated with spatial attentional in the processes in the opposite hemisphere. And this is, so they do make this big sweeping claim, then whatever's going on attentionally in the left hemisphere is pretty much the same as the right hemisphere, just not dominant. But I think you need a little bit more information to make this claim. First off, pretty low number of patients, only 11, I think, especially with given the really massive diversity in lesions which can cause neglect, you need a bigger sample to really be confident about this. And you actually need to do some quantitative comparisons between hemispheres to see what's going on comparatively and see if we can actually pull out differences between the two. And that leads us into the aims of this study. So purposes of this study is to identify the neurocorrelates of left and right egocentric and allocentric neglect in a representative sample of stroke survivors. And this is the VLSM component of this analysis. And then move on to the secondary aim, which is to determine whether right neglect is supported by left hemisphere homologs of regions which are critical for left lateralized neglect following right hemisphere damage, which is a bit of a mouthful, but I'll do my best to simplify that later on. So we did this in a massive sample of acute stroke patients. There were 499 people we recruited who all did the Oxford Cognitive Screen's Hearts Cancellation task in the acute phase following stroke, and also had available clinical neuroimaging, which I could use to make all these lesion masks. And we ended up with a nice number of patients in each group. So our minimum impairment group was 28 for both allocentric neglects, but within each of these comparisons, we also included these 283 neglect controls, which you'll see at the bottom row of this diagram, meaning that pretty much every analysis we ran had at least 300 patients in it, and we're looking at both sides of the brain. So on to VLSIM. So VLSIM stands for voxel lesion symptom mapping, and it's a pretty common neuropsychological approach. The figure illustrates the general pipeline for this analysis. If you have any questions about exactly what we did, I can go into it in more detail. But we ran 4 separate VLSIM analyses looking at the neurocorrelates independently of left egocentric, right egocentric, left allocentric, and right allocentric neglect, all run independently. And importantly, we did these groups based on behavioral grouping. So you fell in the left neglect group if you had left neglect, regardless of where your lesion was. So we're not grouping patients according to where their lesions is, we're grouping them according to their behavior. We did voxel-wise comparisons, minimum overlap of 10, so only looked at voxels with at least 10 lesions in them. Given that we binarized impairment, we used a non-parametric Liebermeister measure as our actual statistical test, use this non-neglect group as controls, and use Bonferroni corrections, which is worth pointing out because Bonferroni corrections are pretty widely regarded as being too conservative for VLSM, but we wanted to be as conservative as possible for this. So next state of the analysis is the lasso bit. So lasso stands for least absolute squared selection operator, which is pretty much a fancier way of doing stepwise linear regression. So in this kind of analysis, you throw in a bunch of covariates which could potentially explain an impairment. Use lasso to select which ones you're actually interested in and then go from there. So in this study, what we did is any ROI defined based on a couple of atlases that actually contained significant voxels in the VLSIM analysis was considered for a potential covariate. We used lasso analysis to select which ones were actually explaining most of the variance. We evaluated the fit of these models by looking at how percentage damage to these areas actually predicted binarized neglect impairment versus a null model of just lesion volume. And then we did the core of this analysis, which is this hemisphere swap, in which we take the model which we made based off of the right hemisphere data, apply it to the left hemisphere data to try to see if the homologs actually predict impairment, and run some tests to see whether or not that's good. So first off, our VLSIM results. This is a little small. I hope you can see it well. I'm going to do my best to point things out. So first off, I'm not going to talk very much about the results for left egocentric and left allocentric because these highlighted in blue agree really well with past literature. So it's a nice finding because it shows that our data is actually good. It's pulling out what it needs to pull out, but not super theoretically interesting at the moment. What is interesting, however, is neurocorrelates we get for right allocentric neglect, which is highlighted in red, right down here, and this falls in the anterior division of the parahippocampal gyrus within the left hemisphere. And this is quite nice because the parahippocampal gyrus is an area that's been traditionally associated with object level processing, particularly within the ventral visual stream. And so it conceptually makes sense. And this is the first study that's ever managed to find a neurocorrelate associated with right allocentric neglect. So this is a cool new finding. The other bit, which is really interesting, is the neurocorrelate we found for right egocentric neglect, which I think you should be able to see it, but it's this tiny little yellow dot right here, which is interesting. So it's in the parietal operculum cortex, and it's ipsilesional, which is really fascinating. So this is a right hemisphere correlate of right egocentric neglect. This isn't entirely new, so there's been a couple case studies that have found right neglect following right lesions, but it's never been done on this scale before. And within our sample, we had patients with both left and right lesions and right neglect, but the actual driving correlate that the VLSIM is pulling out is ipsilesional, which is really fascinating. So on to the lasso analysis. So what we get while looking at egocentric neglect is we see that the left hemisphere homologs of the ROIs implicated in left egocentric neglect were not found to act as significant predictors of right egocentric neglect. What this means is whatever is going on in the left hemisphere is not the same as whatever is going on in the right hemisphere. And again, you see a nice support for that argument by seeing that the correlate underlying right neglect is actually ipsilesional. So that makes sense. But we see the opposite thing when we look at allocentric neglect. So the model which we built based off the right hemisphere data did actually significantly predict the impairment within the left hemisphere as well. So whatever's going on, object level processing seems to be analogous across hemispheres, but whatever's going on, self-centered processing seems to be completely different, which is interesting. So a couple quick things to run through. So first off, the identified neurocorrelates of left egocentric and allocentric neglect agree with previous literature. This is nice because it shows our data is doing what we actually wanted it to be doing. The study is the first to identify the neurocorrelates of right allocentric neglect. And again, conceptually makes sense. It's quite interesting because it can help guide people to know who might actually be having this problem. Right egocentric neglect was found to be most strongly predicted by ipsilesional damage centered within the right parietal operculum cortex, which is again quite a novel finding and is pretty important to consider in the context of this current theories of hemisphere dominance and spatial attention. So traditionally, the right hemisphere attends to both sides of space, the left hemisphere just attends to contralesional space, which is why you're supposed to get neglect just after right hemisphere damage. But we can see that doesn't necessarily fit the damage, the description. You can get right neglect following both left and right lesions, and we need a bit of research to understand why that's actually happening. I think the core big thing to take away from this is that right egocentric neglect cannot accurately conceptualize as resulting from damage to left hemisphere homologs. Whatever's going on in the other hemisphere is different, and it's really important to acknowledge this when doing research with different kinds of neglect. Important takeaway, if I could take one thing away, is that neglect is really, really complicated. It's not just right hemisphere damage, left neglect. And if you're going to do any research involving neglect, you really need to split it apart into these separate behavioral groups. So other research has shown they're dissociated, they follow different recovery trajectories, they're differentially associated with long-term functional outcomes. There's really are a lot of differences between these neglect subtypes. And so it doesn't make sense just to say all patients with neglect. If you want to do neglect research, break it down into different groups and then go from there. And then again, that this idea of right neglect just being an anatomical homologue of left neglect isn't correct. Something different is going on there. I think from what we've seen, it's probably best to think about right neglect as kind of a common symptom of multiple underlying causes rather than this clean unitary syndrome. So quick thanks to everyone who helped out with this. So these are my two co-authors, and Nayla's my supervisor, my lab, and then the people who give me money to do research. So thank you to them. You can look me up on Twitter if you're interested, hunt down my lab website. And also, because I'm a big fan of open science, pretty much all the data for this study is openly available. I think all the code is there, all the lesion voxel significant maps, all that stuff is openly available. And this manuscript is under review, but hopefully it will be out in the world pretty soon. Great, thank you for your time.
Thank you, Margaret. Good luck for the revisions of the paper, and thank you from the whole community for sharing your data and your code, because it's an impressive data set.
Yeah.
If I can start off with a question, again, biased by my language research, do you think language lateralization can play a role at all in how attention gets specialized on the left versus the right hemisphere?
Yeah. So the paper I talked about, the second one I talked about from Karnarth, their idea is that spatial attention in the left hemisphere is held by these like relics of old spatial attention areas that are now taken over by language. And so one of his ideas is that we don't actually it's not dominant in the left hemisphere because those areas have been taken over by language. So that would mean that the side language is on does actually affect which side of the brain affects what's paying attention to where. But I'm not entirely sure. That is their idea.
And in your sample, you don't have enough, like you don't have data on the language lateralization, and you don't have enough left-handed to try to put it as a covariate?
We don't have data on language lateralization, but we do have like standard neuropsychological language screening assessments. So that could be put in at some point in time.
That would be very interesting. I don't know, we have like, I think 1 minute if all the speakers want to come back on screen, if anyone has any last questions. If they come right when we stop, we will be able to send them to you if they come in the chat or in the Q&A. But yeah, otherwise I'm gonna just thank you very much for the 3 brilliant talks, extremely interesting. Do motion if you have questions. Yeah, exactly.
Hi. So yes, fantastic talks, everyone. Margaret, I had a question regarding the neglect syndrome that we were talking about. Has their work been done on other modalities of recordings, such as EEG, MEG, stuff like that, as well? And what change have they seen? Is it changes on the ERPs, for example, that are kind of in line with the old research on neglect that was done?
So I think it's quite tricky to do any kind of functional imaging with neglect because a lot of neglect recovers within the first 10 days following stroke, and it's really difficult to get access to these patients right then. And a lot of these patients are old people who can't go in scanners, can't focus for long enough, have other stroke deficits. So I don't think a lot of nice neglect imaging work has been done, but there has definitely been some functional, specifically fMRI, but I'm not all that familiar with it.
Okay, thank you.
Yeah, maybe just one last following this. Do you think that maybe the left cases are sort of neglected because they have predominantly language deficits? So we just neuropsychologically we focus on those deficits instead of investigating the visual spatial.
Yep. So I've actually done a lot of work on how neglect is assessed in a clinical environment, and there's huge expectation biases in people who have neglect following left hemisphere lesions because you traditionally wouldn't expect that. And so even if the neglect is severe and behaviorally apparent, it doesn't get diagnosed observationally because that's not what clinicians are expecting to see. So another reason why neuropsychological assessment is really important because it can keep us from being biased humans.
Thank you very much. This was a great way to start the morning here on the East Coast. And we are going to have now 10 minutes of break before the next session, and keep following on Twitter the development of the conference. And yeah, thank you again.
Thank you.
Bye.
Thanks everybody for joining in. I will end the talk in 10 seconds.
Okay, hi everyone. Thank you all for joining us. We have a super interesting lineup of speakers. We talked about neural processing and perception of speech. We'll start with our first speaker here, Dr. Sang-Gu Kim, with the title Neural Encoding of Phonemes Modulated by Linguistic Information. Dr. Kim is a postdoc at the Department of Psychology and Neuroscience at Duke University. Just to remind you, let me just a second share my screen for 1 second. Sorry, okay, I don't find it now. You are welcome to ask questions in the chat box. I will read it out loud once the talk is over. Dr. Kim, you can start.
All right. So, okay. Right. Thank you for the introduction. So today my talk is about neural encoding of phonemes, and this work is being done in collaboration with Federico Di Martino from University of Maastricht and also my PI Tobias Ross. And this work is still in progress, but I like to share some interim result with you and get some nice feedback. Right. So our group has been working on the temporal processing of speech as it is crucial in transforming acoustic information to linguistic information. And previously we used a dedicated scrambling algorithm to control the temporal extent of speech structure in which the sequence of segment is selected so that the transition between segments would be similar in the scrambled and the original speech in terms of co-choreogram representation. So this process minimized the acoustic artifacts from scrambling while disrupting the temporal structure of speech. So using this Quilting algorithm, we presented speech stimuli with varying intact temporal extent, and we found that the bold activation in the bilateral superior temporal ossify increased as a function of segment length. And in a subsequent study, we also observed that the left inferior frontal gyrus also showed quite similar response curve, only for a familiar language, which was English, but not for an unfamiliar language, which was Korean. Right. And recently, the encoding of natural speech in fMRI time series was demonstrated. So this motivated us to investigate how the temporal and linguistic context effects that we previously found would modulate such an encoding of speech in fMRI data. To address this question, we used the same bilingual, so Korean and English stimuli from the previous study in a 2x2 factorial design. So 2 levels of language, English and Korean, and 2 levels of quilting or temporal extent. So the original speech and then very short phoneme quilt. Here I have an example for English phoneme. And this is Korean phoneme. So you might have recognized some English phonemes, but the temporal structures are clearly disrupted. So using this stimuli, we acquired multi-band API data at 3T from 10 native English speakers without any knowledge in Korean, with a small test to maintain their attention level. And we repeated 3 sessions of 8 rounds, resulting in 205 minutes of data per subject. And we performed a linearized encoding analysis, which combined the FIR modeling and rich regression. So in the FIR modeling, you estimate the both signal from the current and the previous features from the current and previous time points. And the features were envelope and 4 different phoneme classes based on the manner of articulation. And for rich regression, we used rich trace method to optimize the regularization. And the model was validated over 2 folds. Right. And then to infer the significance of each individual feature or condition, we use a model comparison. That is, we create a different null models which lacks some of features or condition and compare the prediction accuracy, which were measured by Pearson correlation, and then the significance was inferred at the group level using a cluster-based permutation test. And here I'll highlight these 3 comparisons that were most relevant to the main focus of our study. So the first one, the effect of phonemes. So we compare the prediction accuracy of a model where you have 4 phoneme classes and 1 envelope feature, and another model with only envelope feature. And then the question was whether addition of phonetic features would increase the prediction accuracy. So these were individual Pearson correlation differences across different subjects, and this was a group level t-static map. We found increase of prediction accuracy in the left STS, which indicates the encoding of phonemes in this area. And we also found decreased prediction accuracy in around Hessel's and Temporal bilaterally, which suggests the phonemes were not informative in this area, but the envelope that was actually informative was penalized together with the phoneme, so it reduced the prediction accuracy. And then for the effect of temporal context, we compare the full model where we predict each condition separately. So for English phoneme, original, Korean phoneme, and Korean original. And then with a reduced model where you don't, or you pull the original speech and the phoneme quotes together. So there is only distinction for the language. And then having different predictor for different language improved the prediction accuracy in the left STS again, and also decreased the prediction accuracy in the Hessel's and Temporal. So this suggests that in the left STS, the speech was differently encoded in the original speech than the phoneme quilt. And so this is the effect of linguistic context. So we compare the full model again, and this time the reduced model was pulling English and Korean together. So there was only distinction between the original speech and the phoneme quilt. And we also found a similar effect. So the speech was differently encoded between languages in the left STS. So here, white outlines mark the p-value less than 5%. And it wasn't in the Hessel's and Temporal. So to summarize, we found that rapid features of speech were encoded in the ephemeral time series. In particular, the acoustic feature was encoded in the primary and surrounding auditory cortices, whereas the phonetics features were encoded in the left temporal sulcus. Interestingly, the encoding of speech was modulated by temporal and linguistic context in the left STS at group level. And we expect to find more interesting effect at the individual level, such as an interaction between context and the inferior frontal gyrus with some different lateralization across the subject. And the decoding analysis may result in additional insight on the spatial pattern of the neural responses. I'd like to thank the collaborators again, and also acknowledge the funding source from the NIH, and thank you for your attention, and I'm happy to take your questions.
Thank you very much, Dr. Kim, for your wonderful talk. Before we'll ask, while we're waiting for questions, we didn't hear the demo, unfortunately. Could you reshare or share the audio and then try again to play these demos?
All right. Let me try that again. So can you hear this clicking sound? Okay, so this was the, all right, so this was the English phoneme quilt. Right, and then now this is the Korean phoneme quilt. Yeah.
Okay, now it works. Thank you very much.
Okay.
Let me ask you a question. So you showed us the encoding. Do you also plan to do decoding? Speech of the streams that you just played, or
yeah, so we are kind of considering a decoding analysis, probably differentiating for different conditions would be first. And also we are thinking whether it would be possible at more fine time scale. But I mean, to begin with, probably we might try to differentiate for different condition or different language or different temporal extent from the signal. And yeah, probably, I don't know. I mean, so we expect to find also similar distinctions. So this language or temporal extent to be informative would be informative in the left STS and some other higher order area, but not in the primary auditory cortex. Yeah, that's our current expectation.
Okay. Thank you very much. And we're moving to the next speaker. The next speaker is Prachi Patel with the title Neural Modulation to Direction and Speaker in Spatial Multi-Talker Speech Perception. Prachi is a first-year PhD student with Professor Nima Mesgarani at Columbia University. She studies the cortical encoding of spatial speech and music using intracranial recordings in humans. Prachi?
All right. So you see my screen well? Yeah. All right. So today I'm going to talk about how the brain is modulated when the listener attends to a direction of a talker versus when the listener attends to the pitch, for example, of the talker. So up until last year, we were able to attend poster sessions in person where we could focus on the presenter's voice, regardless of the crowd in the background. And if we get bored like my sister over here, we could switch our attention to the neighboring speaker. We could listen to their voice as well as their content. So the questions I'll be addressing today are, what is the representation of spatially separated multiple talkers in the brain? And how attention to direction of the talker versus attention to the voice of the talker modulates the brain responses. So to answer these questions, we recorded intracranially from 7 epilepsy patients, and the coverage in the brain included 32 electrodes in Hechel's gyrus, which is the primary auditory cortex over here, 25 in planum temporal, which is this intermediate region over here, and 53 in STG, which is the higher order auditory area over here. And for the analysis, we look at the high gamma response of these recordings. So we started simple. We looked at how a single spatial talker was first encoded in the brain. specifically how the direction of this talker was encoded in the brain, as well as how the speech content of the talker was encoded. And the task we had a male and a female talker. Male could be on the left side of the listener or on the right side. Female could also be on the left side or on the right side of the listener. And for the speech itself, we had 50 American English sentences, which were on an average 5 seconds long. And the questions we asked from the brain recordings were, how is the talker direction changes these recordings, and how is the talker's speech modulating these recordings? So before I dive into the results, these are the two important terms I'll be using a lot in the results. So I'm just introducing them over here. Contralateral means opposite. So if you're recording from the right brain, the talker is in the opposite or the left direction. And ipsilateral means same. So if you're recording from the right brain, the talker is in the same right direction. So moving to the results from a single talker scenario, if we look at direction encoding, let's see what happens in the brain responses. So here you have average brain response from all the recording sites in the brain. The x-axis is time, so the recording was done over time. And the y-axis is the amplitude of the response. These are the responses to consonants from the speech, and the pink part indicates the response to contralateral talker. So recording from the right brain, the talker was on the opposite side. And blue part represents when the talker was on the same side as the recording site. So you can see that when the talker direction changes, the shape of the brain response doesn't change, but the mean is rising. The mean shifts based on where the talker is. If it's on the contralateral side, the mean is higher. On the ipsilateral side, mean is similar, like lower. And the same thing happens for vowels as well. The shape doesn't change when the direction changes, but the mean shifts based on the direction of the talker. So the direction of the talker was modulating the mean response level in the brain. And next we look at what was happening when the speech was being represented in the brain. So the question was, when you hear a phoneme, say sa, from the left side versus right side, is it equally well encoded in your brain regardless of which side it is coming from? Or do these electrodes prefer to encode phoneme only when it is presented on the contra but not on the ipsi? So we wanted to see those differences. And in the results, we find that the phonemes are equally well encoded in the brain, regardless of the direction of the talker. So even when the talker was on the contralateral side or the ipsilateral side, the phonemes of that talker were equally encoded, shown by the overlapping graphs over here. So the neural encoding of phonetic features was unaffected by the talker direction. So these are like the two main findings from the single talker scenario, where the direction of the talker was changing the mean in the brain, and the neural encoding of phonemes was equivalent regardless of the talker direction. So moving to a more complex multi-talker scenario where you have a male and a female simultaneously talking, but they're separated in space. And the conditions we had were male being attended on the left side, male attended on the right side, female attended on the left, and female attended on the right. And they were the same 50 American English sentences, 5 seconds long on an average. Now in this setup, we're going to ask two main questions. The first question is, just by the presence of these two talkers in space simultaneously talking, what is the encoding like in the brain? So it is a bottom-up stimulus-driven effect. And the next question is, in this encoding that was present in the brain, if you attend to left versus right, what changes in this encoding? And if you attend to male versus female, what changes? So those are the top-down attentional effects. So we'll be looking at attention to direction and how that changes this encoding, and how the attention to talker, male versus female, changes this encoding. So first, looking at the bottom-up effect, in the same tasks, to analyze this data, we're going to compare everything in the blue over here, combined versus everything in the yellow over here. So we are basically looking at all the speech coming from left side versus all the speech coming from right side. So here they're like combining these attention conditions to make sure that this is a bottom-up representation in the brain. So just to refresh your memory, this is what we find in single talker case where the phonemes were equally well encoded regardless of the talker being on the contra or IPSI side. Here the graphs are overlapping. So the phonetic encoding is equally well regardless of a direction of talker for a single talker case. But surprisingly, when we do the exact same analysis for multi-talker case, we find that the brain was preferring phonemes of the contralateral talker. Meaning, if you have two talkers simultaneously talking in space, your right brain is going to encode the left talker more, and your left brain is going to encode the right talker more. And this automatically happens in your brain. So neural sites preferentially encode the speech of the talker on the contralateral side. Looking at the effects of attention now, first we focused on what happens when you attend to the direction. So if you attend to left side versus right side, what changes in your brain? Again, to refresh your memory from the single talker, we found that attention to contra direction versus IPSI direction was changing the mean in the brain. And like as expected, this is what we find in multi-talker. So when you have two talkers simultaneously talking, attending to the contralateral talker versus attending to the ipsilateral talker is also shifting the mean in the brain. So you again see that attention to contra is giving a higher mean response, whereas attention to IPSI has relatively lower mean response. This is for the consonants, and the same thing is observed for the vowels as well. The shape doesn't change, but attention to direction is changing the mean in the brain. So attention to talker direction modulates the mean response level of the neural sites. And finally, we looked at what happens when you attend to talker's voice, meaning if you attend to male versus female, what changes in your brain. To set up a baseline here, we subtracted the sound, the acoustics, the spectrograms of female minus male to generate this acoustic profile from the stimulus itself, where the male pitch is dominant in the blue part over here at lower frequencies, and the female pitch is dominant in red parts over here. Now, when you do the analysis in the brain, we find something similar. For the exact same mixture, when you attend to the male talker, you see that there is a peak in response in the brain responses, showing that the electrodes are tracking the male pitch when the male is being attended. And when you switch your attention to the female in the same mixture, you'll find peaks in red over here in the brain responses, which correlate with the peaks in the acoustics, meaning the female was being tracked. And we quantified it over here by taking the first principal component across the brain data to find that the brain profile in the brain frequency profile in blue is highly correlated with the acoustic profile in pink. So attention to talker's voice was modulating the spectrotemporal receptive field of the neural sites. And finally, we looked at what is the relationship with latency of these attentional effects. The reason why we do this is because as you move from ear all the way to the auditory cortex, the latency rises because of the hierarchy. And even in the auditory cortex, when you move to the deeper structure to a more lateral structure, the latency of the response rises. So we wanted to see how these attentional effects are dominant in terms of latency, meaning where in the hierarchy these effects are dominant. So first, when we look at where the attentional modulation to talker was happening, its relationship with latency was found to be a positive correlation, meaning higher up you are in the auditory pathway, more sensitive you will be to attention to talker. So attentional effects to the talker are almost at the lateral surface of the brain. But surprisingly, we did not find such an effect for attention to direction. This suggests that attention to direction happens much earlier in the pathway. So attention to talker appears in the later stages of the auditory pathway, and it improves with the hierarchy. Attention to direction happens in the earlier stages, and it doesn't improve with hierarchy. So just to summarize, bottom-up stimulus-driven encoding of speech, we found that in the single talker, the brain didn't care where the talker was in space. The phonemes of that talker were equally well encoded. But in a multi-talker case, presence of two talkers, the brain started to suddenly care more about the contralateral talker's phonemes as compared to the ipsilateral talker's phonemes. And effects of attention to direction was modulating the mean of the responses, and attention to the talker modulates the spectrotemporal receptive fields. And I want to thank my lab members, specifically my advisor, Professor Nima Miskarani, our clinical team for this beautiful brain data, Dr. Ashish Mehta, Dr. Jose Herrero, and Dr. Stefan Bickel, and my peers from NAPLAB for their discussions and feedback on this work. We can take questions.
Thank you very much, Prachi. I think you have really compelling and convincing results there. There is one question here. Did you have a lower effect in the signal mean shift when comparing IPSI or contralateral responses, I mean, IPSI versus contralateral responses in the multi-talker condition, or was this effect similar for multi-talker and single talker conditions?
That's a very good question. I find that the effect was, like, in terms of values, it was smaller in multi-talker as compared to single talker. But if you look at correlation of all the electrodes, how they were performing in single talker versus multi-talker angle detection, there was high correlation. So in terms of values, they were smaller in multi-talker, but they were highly correlated based on how sensitive they were to direction of a single talker.
Okay, are there any other questions? You're welcome to type them in the chat box. Otherwise, we will continue to the next speaker. Next, we have Dr. Axel Kalkus, with the title Neural Processing and Perception of Speech in Children with Mild to Moderate Sensory Neural Hearing Loss. Dr. Kalkus has recently taken a faculty position in Brussels, and this study was conducted while she was doing a postdoc in UCL London. Yeah, let's hear it.
Excellent. Well, can you hear me? Yeah. So thanks very much for your interest in my work. I have to confess that I did write the title before actually looking into how long the talk should be. And so just in the interest of time, I've decided to focus on the processing part of this project. So when it comes to hearing impairment, we know that the structure and function of the auditory system is dependent upon stimulation received during the maturation, and that's for hearing loss that are severe to profound. And most of the evidence we have so far actually comes from deaf animal studies, which have shown that in animals, we see changes in the anatomy, in the physiology, and in the functionality of the auditory pathway. Interestingly, in hearing impaired humans with severe to profound hearing impairments, we also see changes such as degeneration of spiral ganglions in the neurons of the cochlea, reduction of volume in the neurons of the cochlear nucleus, and functional reorganization at the level of the auditory cortex. In contrast to the literature we have in regarding severe to profound hearing impairment, much less is known about mild to moderate hearing impairment. The little we know comes from studies conducted on individuals with conductive hearing losses, in most cases due to otitis media with diffusion. And so that gives us indirect evidence about the effects of a mild or moderate hearing impairment on the development of the auditory system. And even in those cases where there's only a temporal mild or moderate impairment of, a temporary mild or moderate hearing impairment, we do see changes in the structure and the function of the developing auditory pathway. However, so far, very little is known about sensory neural, so permanent congenital hearing impairments when they are mild to moderate. Yet it has a high prevalence. And also, even if children with mild to moderate sensory neural hearing loss have residual hearing, the benefits, they are at higher risks of showing language difficulties and auditory processing disorders. So initially, the project that I'm gonna tell you about right now was conducted to answer this question: how does a peripheral impairment affect central processing of sounds in children with mild or moderate hearing impairment? And a good paradigm to answer this question is to use EEG, and specifically what's called a nutball paradigm, where you present a standard sound that's repeated over time. For example, in this case, the standard was ba, and it's interleaved with deviants that are more or less obvious. In this case, there was two types of deviant, one that was very, very obvious and another one that was a lot more subtle. And I'm not sure it's going to work, but let's try. This would be an example. I'm not sure you can hear things. So it's a succession of ba, and then from time to time you have a da that's even very different or mildly different. Interestingly, what we see when we look at the ERPs, so the scalp recorded EEG to these sounds, this is what we see in response of the standard. So this is the average of 600 standards recorded in a 14-year-old child with normal hearing. And what you have to look for are these 4 peaks that are called automatic or obligatory because they're always there. And we're not very original. So the first one is positive, it's called P1, etc., etc. That's the typical response you see in an adolescent. Again, very typical response to the deviants, to the large deviants, where you see a difference, a difference in amplitude, so a larger negativity from around 150 milliseconds to 300 milliseconds, more or less here. And this larger negativity evoked by the deviant compared to the standard is called the mismatch negativity. It indexes neural discrimination, if you want. Interestingly, when you look at the responses evoked in younger children, this is what you get. So this is the typical response in an 8-year-old, and it's perfectly normal. It's very different than the teenager's response. But in younger children, you only see 2 peaks. They're bigger. And ultimately, in between those two, so those two are going to evolve to be a P1 and an N2. And in between those two, we're going to see the N1 and P2 coming, but it's not here yet. But still, when you present those children with a deviant, you do also see a mismatch negativity. So in a way, we can say that we see this neural index of discrimination already in 8 years old. Interestingly, we've used this paradigm to study speech discrimination, neural speech discrimination in children with a hearing impairment, which was only mild or moderate. I know it's a bit dense, but bear with me. The top row are the controls, and the black lines represent the response to the standard. The gray lines represent the response to the deviant. The dotted line is the response to the small deviant, but basically that failed, so we can forget about it. But we can focus on the plain gray line that represent the response to the large, very obvious deviant. And as you can see, at least in controls, so in the upper row, we have a significant portion of the response that was an MMN. So it was significantly different in response to the deviant than the one observed in response to the standard, perfectly normal for their age. And when the children grow older, so on the left part of the plot, you have the younger kids, they're aged 8 to 11. And on the right part of the figure, you have the older children, they're aged 12 to 16. So in the older children, the morphology of the response has changed, but still you see an MMN, everything is in place, great. In the bottom row, though, you still have this distinction between younger and older children. And interestingly, in the younger children, you do have an MMN. It is significantly present for a long portion of the time. It's different, the morphology is a bit different than the one evoked in controls, but it's present. The crucial point here is the bottom right panel, where older children, so 12 to 16-year-old children with a mild to moderate hearing impairment do not show this MMN evoked by speech. Now, one thing I didn't mention is that each of these 4 panels represent about 25 children. So in total, we tested about 100. Interestingly, I actually joined the project later, after this initial phase of testing, precisely 6 years after. So we could call those younger children back and test them again in a longitudinal fashion. So that's, sorry, I'm going to skip that. That's what I'm showing here. So now the disposition is different. The top row show the responses in hearing impaired children. And this is the within individual comparison between children when they were younger and 6 years later. So that's the top right sub-panel, where they're actually older and they're on average 15, so they're right in that 12 to 16-year-old window. And so what we can see is that, okay, there's still a significant portion of the MMN, but it's a lot smaller, and you can see that the morphology is completely different. So even within subjects, there is a change that we could confirm, indicating that just a mild or moderate hearing impairment impacts and has long-term effects that would impact the development of the central auditory system. Now, interestingly, in this case, you might wonder, how were the sounds presented? Well, for everyone, all children, the sounds were presented at 70 dB SPL, and the children with hearing impairment were tested without their hearing aids. So of course, it's going to be harder for them, and they're going to be disadvantaged. So in a follow-up study, we wanted to know how much of a benefit children with a mild to moderate hearing impairment got from amplification in terms of the neural encoding of speech. So what I've shown you so far are cortical responses, the P1 and 1, P2 and 2, and the MMN. They're thought to be evoked at the level of the auditory cortex and later in the auditory system. But of course, we can also look at lower levels of processing in the auditory pathway. And there's one response that's particularly interesting for us, which is the frequency following response, the FFR. It's called that way because it phase locks precisely to the frequency of the sounds that you hear up to a certain limit. And it's thought to be elicited at the level of the inferior colliculus, so at the brainstem level. So what we did here is testing about, well, the sample here is a lot smaller. So we only had about 18 children with a hearing impairment, mild to moderate, and 15 controls. And overall, they were aged 8 to 16 years old. On the right, you can see their audiograms. So the blue shaded area shows the response, the audiograms from the controls, and the orange lines are the audiograms from the children with hearing loss. We used a nutball paradigm. The stimuli were presented at 70 dB SPL. And again, the children with hearing loss were tested without their hearing aids, but this time, only them, not the controls, they heard the sounds twice. They heard them once without any kind of amplification, and a second time, it was randomized, of course, a second time with a simulated amplification based on their own individual audiogram. So it's supposed to mimic what they get in real life. So I'm going to show you the responses here, and we're going to focus on the top row. First in blue, so blue is always the controls, and then orange is always going to be the hearing impaired children. So in blue here, you have the response in the controls with a peak. So this is the response at the subcortical level. And because it phase locks to the frequency content of the sounds, we can analyze the spectral content of the EEG response and see that we get a peak here at 220 Hertz, which was exactly the fundamental frequency of the sounds. Then there's a first harmonic here, perfectly normal. So that's what you would expect in controls. What we saw in children with hearing impairments without any kind of amplification is that the peak at the fundamental frequency was a lot smaller, and there was virtually nothing at higher frequencies. Interestingly, when we provide them with amplification, the amplitude of the F0 becomes a lot larger. Actually, there's no longer a significant difference between the envelope frequency following response in children with a hearing impairment when we compare them to controls. So that's really encouraging. Some of you might be interested in this part, so the TFS portion of the response. In short, I'll tell you that it really follows the structure and the spectral content of the sounds. And because there's a sloping hearing loss, we have to compensate more for the high frequencies, and that explains the very large peak that we see here. I'm not going to go into more details about this. What I want you to remember is that without amplification, the hearing impaired children have smaller responses than controls, but when provided with amplification, I could say in very summary that their activity, the amplitude of the activity normalizes. What do we see at the cortical level? Oh, sorry, one more point. This is a scatter plot of all the children's amplitude at F0 and then the first harmonic in the unamplified and in the amplified condition for the hearing impaired children over age. And so overall, there's no age effect, except here in this amplified condition for the children with hearing impairments, where the younger children benefited more from amplification than the older one. And we see an age effect showing that as they grow older, the response grows smaller. At the cortical level, this should look familiar now. So in blue, again, the response in the controls, and we have the P1 here. This is going to be the N2, and this, the difference between this lighter line and this darker line is the MMN, which is significant over this portion of time. As you can see in the hearing impaired children, whether provided without any kind of amplification or with amplification, there was no significant MMN whatsoever. So to summarize, we can say that even a mild or moderate degradation of the auditory input affects the neural processing of speech at both the subcortical and the cortical levels. Providing hearing aids amplification significantly improves the neural processing of sounds at the subcortical, but not at the cortical level, or at least not as indexed by the MMN. And this observation is in line with previous studies conducted in adults and in animals. In terms of the interpretation for this differential effect of the benefit of amplification, my best hypothesis at this moment is that there is increasing multisensory integration at successive levels of the auditory system. So actually, up to the inferior colliculus, where we've seen that the FFR was elicited, the auditory processing is mostly unimodal. But then after that, you have multimodal integration and multimodal processing in the auditory cortex. So as we go higher in the auditory pathway, the sound processing and neural processing becomes more and more multimodal. And so that could be why we see a benefit at lower levels, but not higher levels provided by amplifications. So I guess that's it for me. I wanted to say merci beaucoup. Also, I didn't find easily a picture of my collaborators that could beat this one, but I want to thank Lorna Halliday and Stuart Rosen for their help on this project, and let me know if you have any questions.
Thank you very much. Before we go on with question, once more, can I ask you to reshare or to share audio and to replay the demo?
I'm not entirely sure it's going to work better, but let's try.
Hear anything?
Can you hear that?
Play it again.
Yeah, for some reason it's not working, but this would be the standard, so the bar. Could you hear that?
No.
Well, it's basically 3 speech stimulus on a continuum from ba to da. So it's rather, so it was constructed in 100 steps, going from one extreme to the next, so the ba to da. And small, very light and hard to discriminate deviant was somewhere in between. Yeah, I'm really sorry. I can share the sounds if someone is interested. I'm happy to send them over email or something.
It's okay. I think it's easier to imagine than the previous demo of the previous speaker.
For sure.
I have a question. You showed us that there was less cortical effect after the amplification for the hearing impaired. There was, I think, a dissociation between the P1 and N1. So the P1 was more normalized than the N1.
Yeah, you have a good eye. So indeed, when you look at only the response to the standards, there is a benefit on the P1, which is an index of detection. So providing amplification actually helped there, but it didn't change anything in terms of neural discrimination between the ba and the da. So there was no MMN without amplification. There's still no MMN when children are provided with amplification. Actually, I can show you that. So yes, so this would be the P1, and it's a little bit bigger when children are provided with amplification. It's pretty much the only difference that we see. Actually, the N2 would probably also be a little bit larger, but there's still no discrimination between the two, and that's one thing we wanted to stress.
Yeah, and also the P1 looks more similar to the controls.
Yeah,
but the N1 isn't.
I'm not sure there is an N1 at this point, probably because overall, I mean, these are all children together from 8 to 16. And as I've shown at the very beginning, there's this morphological change. So
yeah,
I'm not entirely sure. I mean, for the controls, the N1 and P2 are going to appear, and you can see tiny peaks at 200 milliseconds. I don't know if you can see my mouse,
yeah.
but for the hearing impaired children, I can't see that.
Okay. Thank you very much.
My pleasure.
And I'd like to thank again for the speakers. And that's it.